I'm Patrick Brennan. I'm a Professor of Medical Imaging at the University of Sydney in the Faculty of Health Sciences. What I do most of the day is do research in trying to look at better ways of finding cancer earlier, particularly breast cancer and lung cancer. So I work on a daily basis with PhD students, postdocs, other scientists and collaborators from all parts of the world to try and find better ways of finding cancer earlier. I suppose when I look at the first project I ever did, which was almost 30 years ago, I realized that there is an assumption we all make, I think, that we know almost everything about everything. And I think that's a very false assumption, because the project that I did over 30 years ago demonstrated to me that there was real information that was missing. This project was around looking at a better way of imaging a female when she had come in with after a car accident, and she needed an X-ray, and yet there was a possibility of her being pregnant. How could we X-ray that person in a safer way, and a safer way for the unborn child? And I assumed that this was already known and everybody everywhere was implementing this safe technique, and it turned out nobody had any sort of safe procedure on this. So I created, because very much at that time I was very much on my own doing research, I created a technique through a number of different experiments. I arrived at a technique that reduced the dose to the radiation dose, because radiation is harmful to an unborn child, reduced the radiation dose by about 90% and still produced an image that was acceptable under those circumstances. But it became apparent to me that this was there was so much opportunity to actually find out information and find out truth that could actually make a real impact on people. And after that, I was so interested in what we'd found out and what this sort of change that we were able to make in a relatively short period of time, that I then wrote to lots of universities in Europe and asked them would they take me on as a PhD student. And I sort of had, and two of them responded, and I took one of those offers. And ever since that, I've been fascinated by trying to find out truth, but truth that is going to make an impact on people. I had a superb mentor in a fellow called Les Ramsey, who was one of the senior lecturers in the UK where I was studying. And he was important because he introduced to me the rigour that's required when you're doing science. That it's not a sloppy thing that you sort of wander in and do an experiment. You had to be really controlled and detailed and rigorous. And I think he impressed that onto me in a very serious way. The second mentor, I suppose, would have been my PhD supervisor in Queen's University in Belfast. And she was inspirational, but I remember the first time she sat down with me on the first day I was a PhD student. And she said to me, she says, the most important thing you ever do here during your period as a PhD student is you tell the truth. You never ever look for results or alter results or play with results to give you the finding you think is going to be more exciting or more publishable. You always tell the truth. And I think whenever I sit down with my PhD students on their first day, I tell them the same. Developing the question, developing the hypothesis, I think the first thing in my research, even a step before that, is if you're asking a question, is this question important? You know, so we work very, very closely with the clinicians, senior clinicians across the country and in the US and elsewhere, to find out, because you know, particularly as you get older, you find you've got less time, and if you're going to be doing stuff, you want to be doing stuff that has some sort of importance and clinical relevance. So the first thing we do is really look at topics that are going to make a difference to health in Australia or elsewhere. That we know that if we do find the answer to this question, then it should have an impact, and it's going to impact on so many women or so many people or whatever. And certainly that's not necessarily the only way of doing things, but it's certainly not the only way of doing things, because there are rare diseases that people need to focus on as well. But you know, I've been focusing on diseases that are pretty common, that we can, if they're caught early, have a much greater impact on individuals than if they're caught late. And so I feel that if I can help in that detection at an early stage, then I can make a real difference. So the first thing before even we sort of decide on the specific question is we look at an area. We try and find an area where we can make a difference. So, you know, you can look at, you know, government statistics, you know, cancer statistics. There's lots of bodies that have lots of data about, you know, how many people are suffering from this and what, you know, if you found it earlier, what change it can make. That's the first thing that we do. Then when we talk about the question itself, the biggest mistake a PhD student will make, and every PhD student makes this mistake, is they never think the question is big enough. So they'll come up with a question that is really big because they think they've got 3 years, this is a big thing, you know, we've got to really go to town on this, and it's, you know, we have got to make it big. I find the opposite. A good study always has a really small, precise question that may have sub-components within it, but if it's small and those sub-components are small, we can go deep and we can find out not only what's happening, but why it's happening. So I would say 100% of the time, will always bring back PhD students' ideas to something that's much, much smaller. And I think that's an important thing. I think it's about being clear, you know, write objectives regarding what you aim to do in this study in a very definable way. Everything should be measurable. You know, we talk in a language that's often immeasurable. We say things and we think we can measure the impact by what we've just said, but it's a very poor baseline. We've got to be really precise and measurable about what we say. And I think objectives are really good about that. And hypotheses are, because if you're going to prove or disprove a hypothesis, then you've got to be really precise about what you're going to prove or disprove, otherwise you can't do it, you know. It's very hard to think of an example straight off, but say for example, I want to introduce a better way of imaging the breast. You know, how? It's such a vague question. It's just an impossible hypothesis to prove or disprove. But if the hypothesis was something like, I want to use phase contrast imaging because I believe it will show spiculated lesions at an earlier stage compared with our conventional mammography, then that's much more, it's much more easy to prove or disprove that. So the trick is keep your questions relevant, keep them small and keep them measurable. For me, it's the only way that I can make sure that things that I do are going to be implemented within the clinic is to work with the clinicians. So working in university, as they say in the ivory towers within university, is all very well. We can all be very academic and produce lots of academic outputs. And that's fine for some people's types of research. But for me, as I say, it's important to me to try and make a difference, and to do that, to work with the clinicians, to try and devise questions that the clinicians can see will make a real difference, is really important. It's also important for me to be on national advisory bodies, so I can speak to people in government and so on, so I can find out what they think is important. It gives me an opportunity for me to tell them what I think is important, and that really does happen. You know, I was in Canberra with the Department of Health a couple of months ago, and they were talking about extending the age of screening, and I was explaining to them, you can do that, but you've also got to consider the implications that may have on radiation dose, on cancer risk induced by the radiation, and indeed are this extended population of people going to benefit as well as the original group of people that were allocated to screening. And just for your listeners that don't understand screening, you know, screening is offered usually to a particular population of women, often between 50 and 74, but it's been extended to 50. It's being extended, for example, from 50 to 74. So it was from 50 to 70, it's going from 50 to 74. What are the implications of that? So by discussing, they realised they didn't have that knowledge. So now they've come back to me to look at creating a study that would answer those questions in terms of the impact of extending screening ages to diagnostic efficacy and to radiation doses and radiation risks and so on. And then once they have that, then they will be armed to know whether extending it was a good idea or not. So I think you have to constantly work with the clinicians, constantly work with decision makers, policy makers within the country. That can be within the government, it may be within funders, speak to funders so they then provide the money to allow the research to happen that will then be translated. And it's not just about Australia. We can do, in Australia, we can do, as scientists, we can do marvellous things in other countries. We're working very closely with the Vietnamese government at the moment. We just put in a grant there to try and look at ways of transforming diagnostic efficacy around breast cancer in Vietnam. And as you know, at the moment, the way that cancer is diagnosed in Vietnam is not optimal. You know, we can compare it with our Australian data and data from elsewhere, and we can see that the chances of being diagnosed in Vietnam for a woman are much less than they are in Australia. So we've got to do something about that. So I'm working with bodies, Vietnamese governments and institutes within Vietnam, as well as scientists in different parts of Vietnam to try and do something about that. So it's about collaboration, it's about multidisciplinarity, it's about multi-institutional studies. It's much easier just to do research in your office in your ivory tower, but it's much more impactful when you're willing to engage with people and try and bring people in. And I'm not saying it always works. There are always, not always, there are sometimes rows and disagreements because people see different ways of doing things. But that's one of the things you've just got to accept will happen. I suppose a study that I can talk about is, there's a number of studies I could talk about. I suppose the one that's pretty exciting at the moment for us, and it seems to be getting a lot of interest from the government here at the moment and funders, is around trying to predict women who will get breast cancer. Now this has been around for, people have been predicting breast cancer for a number of years, looking at how many children a woman has had, whether she breastfed, how old she is, does she have dense breasts, does she smoke, does she have alcohol, been putting all these sort of individual things into a model and then trying to predict. But we've come up with a rather less strenuous way of predicting cancer, which seems to be as good, if not better, than what other people have used for many years. And this is by flashing an image up. So for example, if a woman in the screening environment in Australia gets imaged every 2 years, as she does for breast screening, the idea here is in 2018 she's shown to have had cancer. In 2012 her image was normal. Could we have seen something on that image that would have predicted that she would have got cancer in later years? So we've come up with this thing where you flash an image for a third of a second. A third of a second is faster than that. And what we do is you get an impression from an image. It's a bit like walking into a room, there's something inappropriate in that room, but you don't know what it is. There's something odd, this situation isn't right, but you can't actually identify what's wrong. It's called the gist, and we utilize that gist by flashing an image up for a third of a second, getting the gist from the person looking at the image. Is there something wrong with this or not? And what we've shown is by them having this flashed up image, they're able to predict, and this is a normal image with no signs that we can find on this image that something's abnormal, yet the gist will say that this is not normal, and we're able to predict the woman who will get cancer in 6 years' time based on this primitive sort of glimpse of an image. Now for that sort of study, you've got to be very, very rigorous, because if you get that wrong, the implications of it are huge. That we start saying you've got a high risk, so therefore you should be doing this. And you can think of the worry and the anxiety that would impart upon a woman. It's only worth doing that if there is a real benefit. So we've got to get it right. Equally, if we're saying people are at low risk, and it turns out they're not, they're actually at high risk of getting cancer, and we get that wrong, then that's a horrible situation as well. So we've got to get it right. So the science behind that has to be completely rigorous to ensure that we are actually finding out the truth and not some sort of version of the truth. So to do that, we've got to, for example, look at many radiologists, because if we just tested you and you have this wonderful ability to predict what's going to happen in 6 years, you can't assume that every radiologist in the country is going to have the same ability. It might be because you've been doing it for 20 years, or it might be that you've been reading 2,000 breast images for the last 10 years. So we can't assume that everyone else has this ability. So we've got to look at a variety of individuals with variety of experiences and backgrounds, males, females, glasses, no glasses, left-handed, right-handed, whatever variable you want to see. You've got to look at them all to see if you can find out exactly what is, whether this is generalizable or whether it's just peculiar to a small group of people. You've also got to look at a large group of images. You know, we could have just picked up, 30 or 40 images and we're getting this effect. Whereas people are looking at thousands in a year. So we've got to make sure that those 30 images were representative, or 60 images or 200 images, however many you look at, are representative of the total population. So you've got to look at large numbers of images. And of course, all the statistic analysis, like we can make stats say whatever we want them to say, we can exclude stuff and we can get whatever finding we want. But, if you really want to find the truth, you've got to use very rigorous analysis. And then once you've got your results, check there's nothing that sort of could have created those results. Like, was it the fact, if we were able to predict the women who were going to get cancer, was it that there was something else unique about their breasts that would have put them in a higher risk, like height density or something like that? So you've got to outrule all those sorts of things. So it's a very, from start to finish, when you're planning the work, it's got to be rigorous, implementing it's got to be rigorous, but then when you're retrospectively analysing stuff, it's got to be rigorous as well. And then of course when you're writing it up, it's got to be rigorous. So how I represent my data, it really depends on what data you have and what you're trying to do. I find in papers, the papers that I write, that I find that putting your data into tables is a very, very effective way of presenting a lot of data. So if you've got a fairly big study and you want to summarize all the data to show what was happening across all those radiologists, across all the different conditions, across the different types of images and so on, a table is a very effective way of doing this. With graphs, you've got to be, graphs are a beautiful way of hitting an audience in a presentation or whatever very, very rapidly. But graphs can be misleading. I think one of the most misleading types of graphs is a pie graph. You get a pie graph and you see these things, but it's very hard to know sometimes which one's bigger than the other, without the numbers. Whereas I would find a histogram a more useful representation often of proportions within a population. So in terms of presentation, it depends. Like certainly if you've got lots of data, I find tables are very important. But if you're trying to get home a particular point, you want to focus in on a particular type of data, or you're doing a fancy presentation or whatever, then you would often use graphs. I personally find histograms the most useful graph to use a lot of the time. Pie graphs I never use. Line graphs are useful, but often line graphs should have time on the x-axis. And that's sometimes abused because a line between this point and this point implies a relationship, which could be completely two independent things. So it really depends on the aim of what you're doing. But I often say to students that presenting data is probably one of the toughest things that they can do in a paper or a presentation. And it often takes, and I don't know often the answer going into it, which is the best way. We often have to do 5 or 6 versions before we actually work out which one's the best one. And it's the trick in science when you're presenting or writing up stuff. The trick is that you've got to present it in the simplest way you can for the reader. The reader is a busy people. They may have several papers to look at. They may be teaching a load of science students and they have only time to get the gist of a paper or whatever. But you've got to present it so the reader isn't fighting to understand what you're trying to say. So that takes several efforts, I think. So you've got the results is a very, very challenging part of any sort of scientific procedure. and it will take several efforts. When you talk about statistics, there's a number of questions you've got to ask, and this is the biggest mistake that people make, I think, in science, is use the wrong statistics because they haven't asked the basic questions. So the first question you've got to ask is something like, what are you doing with these statistics? Are you actually comparing different groups to see are they different? Or are you trying to show there's a relationship between these groups? So for this, for example, we could have been looking at the difference between the normal and the cancerous images, or we could have been looking at the relationship between this GIST response of the radiologist and the women who get cancer. So we could have been looking at this in a number of different ways. But the key thing is first to ask the simple questions. The statistics is about simple questions, not about the fancy sort of language that puts people off. It's about, you know, what are you wanting to do? What is the question you want answering? Is this group of images different from this group of images? If that's what you want to do, then you go down a certain pathway, okay? The next question you have will be, is this data, or are these data parametric or non-parametric? Which sounds like a complicated thing, but it's really asking you, if you looked at the distribution of data, are some of the values small, some of the values large, and the rest of them are nicely distributed in the middle? Okay, so you get this nice sort of bell-shaped curve. A lot of data isn't, particularly if you haven't got a lot of responses or a lot of values, or you're using numbers that are contracted, like I say to you, how do you like this room on a scale of 1 to 4? I'm forcing your answer into a very small group of numbers, 1 to 4. Instead, if I asked you how old you were, you'd have a much greater continuous curve. It could be anywhere between 1 and whatever, you know, but you'd have far more numbers to play with. You often end up with that non-parametric or skewed data when you've got a limited number of numbers that you're looking at, or you haven't got a large population. That's the second question. So first question is what are you actually asking? Second question is your data parametric or non-parametric? And then there's a series of other questions that you can ask. Are they paired comparisons? So in this group of images, are these 25 images of normal from the same women, same 25 women who eventually got cancer later on? Or are these different women? If they're the same women, then it's a paired comparison. If they're different, it's non-paired comparison, and so on. And then, you know, and how many groups? Is it 2 groups or more than 2 groups you're comparing, and so on? So there's all these questions you've got to ask. Now, a lot of computer packages now are pretty sensible. They force you to ask those questions. Where the problem arises is where you think you know what test to use and you just use it, and it ends up being the wrong test. And that's a cardinal sin in science, is to use the wrong statistical test, but often you find it. What I try to impress upon my students is you should never expect a result. You know, you may have a hypothesis and so on, but that hypothesis is either going to be proven or disproven. You should never expect a result. You've just got to see where the results take you. And they can lead you down very exciting paths, even though they weren't the path that you originally thought about. Now, I'll give you an example, a study that we did about 2 years ago, or a year and a half ago. We had this if any woman is undergoing breast screening procedures in Australia, what happens is they have their breast imaged, and then these images are read by 2 radiologists independently. So if one makes the mistake, then hopefully the other one will catch that mistake. And this is done in a very sort of convenient way, that you're a radiologist, you read it, and then your mate in the next office reads it. And we thought we could now use a much more, particularly with digital imaging where images can be transferred anywhere, we could have a much more intellectual approach about the pairing. Because there's no point putting one person with another person and these 2 people make the same mistakes. You want to put people together who make different mistakes. So we thought there was definitely an opportunity here for improving things, and it turned out that the random way was just as good as any intellectual way. You know, the fact that you were close to someone, it didn't seem to have any disadvantage. So that was a hugely unexpected result. But what that did show us was that the pairing of the radiologists was supremely better than just one radiologist, regardless of whether it was random or more contrived. And so, yeah, that was a very, very unexpected result. And we got funding for that study and people expected that we were going to come up with a much more sophisticated way of doing things, and we just had to admit at the end there wasn't. Careless scientists are very dangerous people. You know, scientists who will do something very rapidly and they don't go back and they don't check and they don't read. I once had a PhD student from Singapore, wonderful PhD student, Dr. Pauline So, who's now based in the National University of Singapore. And one of her papers, we were looking at characteristics of radiologists who perform better than others. And this is an important paper because it had real implications for the clinical situation. And we published it in the highest ranking imaging journal we could find. And I remember her sending me a version, I'd send it back. We'd start with the introduction, send her, well, in fact, we didn't start with the introduction because I find the best way of a sequence of a paper is start with your methods. After you've done the experiment, start with your methods, get that down, then do your results, then do your discussion, then go back to the introduction. Because if you've done all that, you then know why you did the study. And rather than, you know, writing why you did the study before you've even seen the results, it seems a strange way, because the paper has to be a story. You've got to sell this exciting sort of episode of science to people, but you've got to tell a story. So if your discussion and results are saying something entirely different from your introduction, you've lost the reader in the first couple of pages. So we had this, and so she sent me a little bit of the methods and I sent it back and the results. And I deliberately, as an experiment, counted how many versions of that paper we had, and we had 102. Before that paper went to the journal for the first time, we had 102 versions of that paper before we thought it was acceptable. And that's what you've got to do. The days are long gone, if you want to make any sort of difference, the days are long gone for individual disciplines just to work, particularly in medicine, if you want to make a difference. You know, you cannot work, an orthopedic surgeon cannot work in isolation of maybe a public health scientist, in isolation of a statistician, in isolation of a health economist. If I find a new way of imaging, the first question I'll be asked, well, can we afford it? You know, it may be a brilliant way of imaging, but before the government will implement it, they'll say, well, what is this going to cost? So I need the health economist there. If you want to make a difference, you've got to work with lots of people, and it's so much more fun, and you learn. The trick is about being a scientist, it's not about being smart, it's about surrounding yourself with smart people. And that's what I've managed to do all my life. Obviously they have different skills, and those skills should be complementary as opposed to overlapping. You know, they have different knowledge, they have different skills, so they do that. But it's about perspectives. They see things in different ways. For example, one group of collaborators we work lots with are consumers. And a consumer in my area is someone who has suffered from cancer. Now I can come up with some really smart ideas, and then I bring them in as a collaborator, as a partner in the research, and we're talking about something, and they say, well, this is going to make, I don't care about this. You know, it's not going to make any difference to me whatsoever. You know, it's not going to change anything that happened to me. But so they will bring perspectives, and they will, or it's the opposite. They say, now, this needs to get out there. You know, all the way you're publishing this in the scientific literature, and you're going to the meetings, and you're talking at presentations. But the consumers don't know this. They don't know to go to the GP and say, well, I want this form of imaging. So, you know, they bring knowledge to a situation which is very, very beneficial. And it's the same with public health scientists. You know, brilliant people. If you want to change things in the public health arena, you know, you've got to be involving these people. So people do see things from different perspectives, and they have different knowledge, and that sort of, and it's difficult. Collaboration, you know, everyone says you must collaborate, and you must collaborate, but it's sometimes not as easy as it sounds. You know, trying to bring people together regularly in a single room alone is a challenge. Well, the first thing about communicating research is I used to head the breast cancer group in the university. And I stood down from that recently, but now I head the cancer group in health sciences. And I know we've got to communicate our findings. There's no point doing good research and nobody knows about it. So we've got our typical academic routes. We want to publish in good journals. No point publishing in journals that will accept you, that are not highly ranked, are not going to be read. You want to get in good journals. You want to make sure the readership of those journals, the people you want to be reading your research, you will have to get out there and present, at the good conferences, national, international conferences. But that only goes so far. It's great for academic outputs, but how is it going to make a difference? So when I was head of the breast cancer group and now head of the cancer group, where I'm going to, what we do is, and what I'm going to do later on this year, is have consumer evenings. And when I was looking after breast cancer, we had groups of consumers that came in twice a year, and the room was packed, far bigger room than this, maybe 150, 200 women, for example, who had breast cancer. They would come with loads of questions. There were a few scientists talk about their research, and this really got out to the public what we were achieving and so on. And that's something that I'm promoting this year within the cancer group as well. So I think involvement with the consumers is really, really important. It's also important to get out there and speak to, again, governments, policymakers and so on. And this isn't, how do you contact someone in government? But it's not that hard. You just, once people know you're doing reasonable research, the doors start opening. And all I have to do is drop an email to the Department of Health, to people I know there, and say, look, we've got this interesting finding. Can we come up the next couple of months and talk to you about it? And it's something you may want to consider. Getting onto these, also getting onto these reference agencies, the screening agencies, for example, or whatever, national bodies, to try and get an opportunity to talk about your research and show why you think it's important and so on. So dissemination of research is not just about the academic outputs. It's much broader than that if you want to make a difference. I think absolutely go for it. I think there are many exciting careers. There are many exciting careers, but science is about, the science is about finding out the truth about stuff, finding stuff out that nobody in this world knows about. It's not written down anywhere. It's finding new knowledge and then finding ways of using that new knowledge to make a difference and making it good for society. Like, there can't be much more useful sort of enterprises than that sort of thing. It's rigorous. It's not easy. There'll be hurdles, but those hurdles are often not so much academic hurdles as practical difficulties that you encounter. So if you're going to do science, I think the skills you need are not necessarily brilliant. You don't need to be brilliant to be a scientist, but you do need to be hardworking. You do need to be pragmatic. You do need to be able to adapt, and you need to be able to tell the truth.